author and 8 co-author papers. And it's a pleasure to have him out. And let's welcome Thomas. And please, let's go ahead.
Thank you very much. It's nice to be out here. New Mexico is a really lovely place, from what I've seen of it over the week. So essentially what I'd like to talk to you about today is what I did for my dissertation project for the last, consumed a little over a couple of years of my life at the Cognition, Brain, and Autism Lab at the University of Alabama, Birmingham. And just to sort of give you a background of what it is that I study and what it is that I'm interested in, most of what I'm curious about is the structure and function of the striatum using MRI. So this is involved in a number of cognitive functions, and it's a very intense and complicated network. When I'm referring to the striatum, I'm talking about this little area right here in my diagram, and it has a lot of anatomical and neurotransmitter connections via direct and indirect pathways with the cortex, the thalamus, globus pallidus, other subcortical structures such as the substantia nigra and ventral tegmental area, and it communicates using a variety of different types of neurotransmitters. The one that it's most famous for is dopamine and D1 and D2 receptors, but it also uses GABAergic, glutamatergic, oxytonergic, so neuropeptide-based communication, and in addition to other non-traditional transmitters like NO. But still trying to get this. Typically, when we're looking at it in the neuroimaging literature, we're looking at two discrete functional regions, such as, by that I mean the ventral and the dorsal striatum. Now, the ventral striatum consists of rostral ventral caudate and putamen, but it also is comprised of an area called the nucleus accumbens, which is highly involved in a lot of different kinds of behaviors, but the one that we sort of associated with most famously is its role in reward and reward processing. Now, it crosses a lot of different domains in reward, such as how motivated you are for that award, or even the planning and decision making that goes into your approach towards that reward, but it can also be influenced by the valence of the stimulus, like how pleasant or unpleasant it is, how salient it is to you, so how much does it grab your attention or otherwise, and it can also respond to a variety of different types of rewarding stimuli, such as drugs, money, food, or even some of the literature suggesting social stimuli. The dorsal striatum, on the other hand, is essentially anything that's non-rostral ventral caudate putamen, and the line of demarcation in the histological literature, so not even the MRI literature, is really vague, so it's hard to know exactly where its split occurs. But the dorsal striatum has some roles in reward as well, but it's primarily associated with motor sequence learning and execution and multisensory integration, particularly tactile or visual stimuli, as it's associated in the caudate. So primarily caudate putamen here, and if you look on the previous slide, this is actually where the nucleus accumbens is located. When you look at large amounts of metadata, when someone is doing a reward task. So we're essentially talking about this little blob in the more anterior part of the brain. But the reason I'm interested in this is the role that it could potentially play in autism spectrum disorder. Now, for those of you unfamiliar with this pathology, it's a pervasive developmental disorder that affects about 1 in 68 children in the United States. Unfortunately, we don't have the data on the adults, which is something that saddens me. But it's primarily characterized by deficits in social communication and interaction, and a history of restricted interests and repetitive behaviors or activities, and that can range from everything from hand flapping to rocking, or obsession with spinning objects, or special interest objects like trains or cars or fish. But the reason it's interesting to me is that a lot of these theories that we see out there in autism spectrum disorder, especially when it comes to brain imaging, proposes that the deficits that we see in individuals on the spectrum come from these alterations in how brain regions communicate with one another. which we typically study in one of two ways, using either the temporal correlations in the bold signal through functional connectivity or the diffusion of water in the brain, which we then try to recreate in the form of white matter tracks. Now, if you look at the narrative of functional connectivity in individuals on the spectrum, you see this sort of a long-range underconnectivity, so more distal parts of the brain show less R values or synchrony, whereas you get this short-range overconnectivity, which means regions of the brain that are closer to one another display higher synchrony or higher correlations, but also regions that are sort of within a single region, like voxels that are neighboring to one another, are more highly correlated with one another. But if you look at the diffusion literature, you see that there's this compromised white matter integrity, and I'm just going to, that's not entirely correct when we refer to anisotropy, but it's used in the literature a lot, is sort of reduced across the board in the individuals on the spectrum. And a lot of what's been posited for those functional connections is that something is wrong with the underlying white matter that is causing these alterations in functional connectivity. But the thing is, we haven't actually done all that many studies that analyze functional connectivity data and diffusion-weighted imaging data relative to one another. And most of what we have done isn't on the communication between subcortical regions with the rest of the brain, it's between cortical regions with other cortical regions, such as, say, frontal cortex to inferior parietal lobule and other similar areas. Now, dysfunction of the striatum in particular has been positive as a source of restricted interest and repetitive behaviors in individuals on the spectrum for a very long time. So if you actually look at these charts here, you'll see the growth of the caudate and the putamen in centimeters cubed by year, and an individual's insistence on sameness score on the autism diagnostic interview, and that's the insistence that something has to be done this way and that it has to be done that way all the time, otherwise they get highly stressed. And we see this across frequently with other types of repetitive behaviors as well, such as the rocking and the hand flapping, but what's interesting is that it's there, but it's not always consistent across the literature. The other thing that sort of caught my attention with this question is this social motivation hypothesis of autism that's been going around frequently. And that's sort of positing that individuals on the spectrum have decreased motivation to engage in social situations, and as a result, they have a communication deficit because they fail to orient to it. Now, they found some evidence for this in both the functional and the structural literature, in that when they have an individual on the spectrum do a task that's rewarding, they do see reductions in bold signal in the nucleus accumbens during these types of reward paradigms. And there's also histological studies that have been done suggesting that the volume of the nucleus accumbens is a little smaller in individuals on the spectrum. But what's not entirely clear in the literature is whether this is domain specific or if it's an across-the-board reduction in reward circuitry, like does this apply to food or money, or is it just social situations for individuals on the spectrum? And the other is the role that attention plays in these processes. Because the way that they argue it is that since it's not motivating, you don't orient to it. But I could also see the inverse, where if you have a problem perceiving it, you're not going to develop this motivation. So it creates this sort of chicken and egg problem, and sort of where do we study this first? And as I mentioned before, you don't see a lot of studies, particularly of subcortical structures in general, but especially the striatum one, which is interesting considering the role that it can have on both of these facets of the phenotype. But from what we do have available, we've noticed that there is increased functional connectivity from striatal structures to somatomotor cortex, which is a little hard to see in this image, so I apologize. And the other is this trend of reduced fractional anisotropy in these exact same regions. So what we're seeing is increased functional connectivity, but decreased white matter structure metrics, which is interesting. And I haven't seen a lot of discussions about this relationship. So to just sort of posit what's out there as far as the way the theory is structured according to the literature, we have communication between various different brain regions using our signal. And in theory, that should be moderated or even mediated by the white matter, underlying white matter. And both of these things should be able to inform us either who is on the spectrum or merely having the diagnosis of autism should be affecting how these develop. But we've got a lot of heterogeneous results. And I have a feeling that there's a reason for that. And we've sort of framed it as, okay, who's on the spectrum and who isn't, but the spectrum can vary. So if I look at an individual person, their social communication skills might be better than, say, an instance of restricted interest or repetitive behaviors. I could look at another participant and their social communication skills could be not all that great, but they could be highly socially motivated and not have many of those repetitive behaviors at all, and so on and so forth. So going in, I wanted to ask a slightly different question. The first thing I wanted to do was sort of take the functional connectivity as we've seen it in the literature so far and compare individuals on the spectrum to typically developing individuals and say, okay, based on everything that's been done before, what do we see in the striatum? But as a second aim of this, I wanted to eschew the group data entirely and focus specifically on behaviors in functional networks related to restricted interest and repetitive behaviors and social motivation. And I also wanted to analyze functional connectivity relative to these anatomical projections of the striatum measured by diffusion-weighted imaging. So to do all that, we went through a lot of different steps. The first thing we had to do was choose a cortical parcellation to use, and I used Choi 2011, Yo 2011, Choi 2012 atlas. That essentially divides the brain up into regions where voxels are highly correlated with one another. So if you look up top here, what they did was they looked at each individual voxel and grouped it with other voxels that it correlated with, basically to the greatest degree, and lumped it into these 7 different networks. And then Choi expanded on that work and placed a lot of subcortical seeds in striatal structures, specifically the caudate, the putamen, and the nucleus accumbens, and decided to group these regions based off of which cortical region they had the greatest functional connectivity with. And they validated at least some of the sensory motor components using tongue movement and motor grasping tasks using MRI. This is coming from Randy Buckner's lab, and now Thomas Yo's lab. And they validated at least some of these connections in primates using track tracing. But this is sort of broken down into 7 different networks: visual, somatomotor, dorsal and ventral attention, limbic, which is really hard to see, but that's essentially amygdala and temporal cortices, frontal-parietal, and default mode network, or resting state brain network. So vision, somatomotor, which is movement, dorsal attention, which is this sort of top-down focused attention, ventral, which is sort of this bottom-up orienting to a new stimulus, like, oh, what's that? And when I resampled this to 2 millimeter space just to get my diffusion data and my structure and my functional data in the same space, some of it was lost to interpolation. So we lost a lot of the visual components. Each of these can be broken down into subnetworks. So somatomotor networks can have 3 or 4 different subnetworks that are part of the same grouping, but they're in functionally distinct areas of the cortex, which means they are essentially part of the same network, but it's a finer parcellation. And I used the finer parcellation for my analysis because I wanted to have a little more specificity. But that's not enough about that. So what we did was we ran Afni's clustering algorithm on all of these different individual networks to generate 107 individual brain regions across the cortex and across subcortical regions. And we were only looking for within network connections, so subcortical regions as they related to cortical regions related to dorsal attention, and subcortical somatomotor regions as they related to cortical somatomotor regions. And to do that, we accessed some open source data from the ABIDE database, or the Autism Brain Imaging Database Exchange, and it contains a very large collection of resting state fMRI, T1 weighted images, diffusion weighted images, and a lot of phenotypic data. But we had to narrow it down a bit with our criteria. So we started out with the 1,044 participants from the ABIDE 2 data set, which is where the majority of the diffusion data is. And out of all of the different sites that were involved with this project, only 319 of those actually collected diffusion data in some form. And of those 5 sites, only 2 of them, specifically New York University and San Diego State University, collected behavioral data for all their participants. So in Institute Pasteur and Barrow Neurological Institute and Trinity College Dublin, they had only collected their phenotypic data, such as the restricted interest and repetitive behavior scores for their patients and not for their typically developing individuals. So after analyzing the NYU and SDSU data sets, we noticed that the vast majority of the participants in the NYU data set did not have a diagnosis of autism spectrum disorder or high-functioning autism. They had something called PDD, not otherwise specified, which isn't exactly on the spectrum. So that left us with just the San Diego State University data set. And after applying our pediatric and intellectual criteria, left us with 55 participants to work with. And we took images from those 55 participants and we pushed them through 2 different quality control pipelines. The first was the Preprocess Connectome Project's Quality Assessment Protocol, or QAP as it's called, which will essentially give you 17 different quality metrics across structural data, functional data, and diffusion data. And we also pushed it through University of Penn's Perelman School of Medicine's Quality Assurance Scripts, which was essentially developed to identify what data metrics could separate good from bad from excellent diffusion data. And they found that using just max voxel intensity outlier counts and temporal signal to noise ratio could differentiate good data from bad data and good data from excellent data. So across all of these different metrics for every single subject, we essentially transformed those metrics to a z-score, and I used that to identify outliers across multiple different domains. In the future, what I'm probably going to start doing is using Mahalanobis distance to identify outliers across either all data types or across individual modes. But once we applied that criteria where we filtered out any participant that had a z-score greater than 3, we tested for group differences and found that the only place where they differed in our quality control metrics was ghosting signal to noise ratio. Now what that means is that areas where there is prone to ghosting or an after image in the MRI signal, in typically developing individuals, there was slightly higher signal in those areas than there were in individuals on the spectrum. But that said, a lot of the SNR values for this data was ranging from, say, 15 to 30. And if you look at the ghosting signal to noise ratio, it was 0.009 compared to 0.007. So this is one of those cases where it's significant, but not necessarily meaningful as far as the strength of the signal goes. We also added a little component to our analysis pipeline where we created a custom template because we were using a pediatric sample. So we took about 332 different brains from the ABIDE 1 and the ABIDE 2 database and applied that same filtering criteria for data image quality. And then we used the remaining images to construct this pediatric template that you see here rendered in FreeServer. Now, if you look on the left here, what you see is the standard MNI 152 template that FSL will use. And overlaid on that here in orange is the pediatric template that we created. So you can see there is a little bit of displacement, and you will actually get a lot of stretching to try to fit a child's brain to an adult brain, which ideally you don't want. So we pushed this through a custom pre-processing pipeline that started with standard realignment, non-warping, and SPM, motion detection using art detect. And then we use ANSYS diffeomorphic co-registration with cross-correlation to register the EPI image to the T1 image, which we skull stripped in AFNI to make it a little easier to do. And then we use segmentation, FSL-FAST segmentation procedure, because there are segmentation procedures that try to segment the brain based off of tissue priors from adult brains. FSL-FAST does not make any assumptions about what those priors are. It just uses K-means classification to try to identify what's gray, white, and cerebral spinal fluid based on the intensity of the image. And from there, we took all of the EPI images in the gray and white matter and CSF and normalized it to our pediatric template using ANSYS diffeomorphic registration with cross-correlation. And from there, we just performed spike regression for motion outliers and checked on motion. And basically any participant that had more than 20% of their time points corrupted by motion, we just didn't use those participants. ACOMP4, which is a principal component analysis-based approach to trying to regress out the effects of white matter, cerebral spinal fluid, and realignment. And we used a slightly different bandpass filter here than is typically used in resting state data, and the reason we did that is that research from Ted Satterwhite's group identified that using this filter, the 0.01, 0.08, will actually reduce the correlations between your functional connectivity and your motion values, which ideally in a pediatric sample is what you want. Another thing that we did, and this is based off of 2 papers that came out just this year, is a few extra analyses specifically analyzing motion and what's called distance dependence artifacts. So when you have someone moving in an MRI scanner, what it'll do is create artificially smooth regions that are a function of that movement, and regions will appear more highly correlated than they actually are, which can generate artificial functional connectivity results. But what's interesting is that in addition to that, that connectivity can vary as a function of distance. So when someone moves in the scanner, let's say this is our seed, areas that are closer to that seed will appear more functionally connected, and areas that are further away from that seed will appear less functionally connected as a function of someone moving in the scanner. Now, if you'll remember from one of my early slides, this is exactly what the field is saying the autism phenotype looks like. So the very first thing we did was to take all the different pairwise correlations that we had across all the different networks we were interested in identifying, and just did Spearman's row correlations with their motion values and the functional connectivity. And we found a few differences here and there, which we corrected for when we did some of our analyses after the fact. But the other thing that we found was that when we took the correlations between the motion and the functional connectivity signal, there was a small but statistically significant correlation with the distance between the 2 ROIs, meaning that ROIs that are further apart appear less correlated, and ROIs that are closer together appear more correlated as a function of that motion. So that is a caveat. So after applying all that criteria and doing all that data cleaning, what we were left with was 38 of those 55 individual participants. We tested them across a number of demographic variables, and the only place where we found a significant group difference was verbal IQ. There's a much greater range in individuals on the spectrum, which is an unfortunate caveat going in. So to sort of transition to the very first aim that we did, we extracted those pairwise R to Z transforms from all 107 different pairwise connections, from all the different pairwise connections within the networks we were interested in analyzing. And for each of those, we performed an Anderson-Darling test for normality on every single FC pair. And any time that that returned normal, we used a t-test. And any time it returned non-normal, we used a non-parametric Wilcox and Rank-Sum test in order to check the differences between the groups, which are FDR corrected for total number of comparisons. For aim number 2, we decided we wanted to use 3 different subscales from 2 metrics that are commonly used to evaluate individuals on the spectrum. So the social motivation score from the social responsiveness scale is a measure of how much you're willing to seek out other people, how pro-social behavior, your confidence in social situations, and is rated on a 0 to 3 Likert scale. But the important thing to remember here is that the greater the score, the more impaired you are in social motivation, or the less socially motivated you are. And the other 2 that we use come from the 6-factor solution of the restricted interest and repetitive behavior scale. And stereotype behaviors is essentially rocking and flapping and rubbing surfaces and covering ears, so basically things that are done almost like tics, with higher scores, again, indicating the more severe or more persistent the behavior. Compulsive behaviors are almost OCD-like symptoms, like checking doors, windows, clocks, and repeating phrases and routines. But again, a higher score means that the compulsive behaviors are more persistent and more intense and potentially impairing. But we wanted to use very specific brain networks in our parcellation to do this analysis. So for SRS motivation, we limited it specifically to these limbic regions that you see in yellow due to their role in motivation and reward. But since attention could be a potentially strong component of this, we also decided to look at dorsal and ventral attention networks. So that's again, focused attention is dorsal, so top down, and ventral is bottom up, orienting to new stimuli. For stereotype behaviors, we stuck almost exclusively to the somatomotor networks that you see up here in blue. So just regions to do with sensory and motor behavior. And for compulsive behaviors, we wanted to use frontal-parietal regions, so areas involved in cognitive control, knowing how to respond to a certain stimulus or when to inhibit one's response to a certain stimulus. So basically, top-down goal-oriented behavior. And as the second part of aim 2, we used DSI Studio to create those using those same different 107 different ROIs to do Q-space diffeomorphic reconstruction in the M&I template space to try to address for group differences in total intracranial volume. And we asked it to generate 1 million tracks per subject. Now, what separates this approach from what we've done in the past in the autism literature when it comes to tensors is tensors will typically fit that eigenvector at the level of a single voxel. But what DSI Studio will do is fit an ODF at that level. Now, you can use traditional measures like fractional anisotropy when you're using the tensor, and you can get what's called generalized fractional anisotropy from the ODF, which is essentially its cousin that's equivalent. But what DSI Studio does differently is it also gives you a metric called quantitative anisotropy. And rather than being this vector or velocity-based function like FA is, quantitative anisotropy is a quantitative value of the number of spins, which can give you a lot more information about where a fiber might be willing to go. So with GFA, you have, this is probably the principal direction. With QA, you have possible direction in maybe 6 different directions, and you set your threshold for where you want it to track. So we set a threshold on this analysis due to the limitations we had with a small sample, in that we set a flag for if any pair of ROIs did not have connections between them for, say, 25% of the subjects. We said, don't do analyses on that. At least 75% of the people in the subject sample have to have pairings between these regions. And from there, we extract the more traditional generalized fractional anisotropy and the normalized version of the quantitative anisotropy from each of those ROI pairs, which we then correlated with the functional connectivity. So to summarize everything, when you look at this dorsal attention network between the 2 groups, so again, this is a top-down orientation. If you look over here on the left where the functional connectivity values are for the y-axis, individuals in white are the typically developing individuals, and individuals in gray are individuals on the spectrum. You see that between regions of the putamen on the left and right side with the anterior cingulate are reduced in individuals on the spectrum. For somatomotor, you actually see the inverse. When you look at the putamen body right around, it's a little hard to see from here because of our eyes is small. But you can see that individuals on the spectrum actually show much greater connectivity with the putamen and the right inferior frontal gyrus and left inferior orbital frontal cortex. But when you get to this frontal parietal region, and this is a subregion of one of those frontal parietal networks I was talking about, you see that individuals on the spectrum display a reduction in functional connectivity between the putamen tail back here with the left IPL and the putamen body and the putamen tail with the precentral gyrus that's located up here. And when you look at the other frontal parietal subregion, which is, again, that cognitive control region, you actually see the inverse, where there's a reduction in where there's an increase in individuals on the spectrum between this region that's sort of left inferior, and middle temporal gyrus with the caudate body. And you see the same thing in this default mode region that's primarily the right temporal pole and the left caudate. That's a little different. Now, moving on to the behavioral correlations, which is the stuff that I was particularly interested in, we see that in these dorsal attention networks, you can see something of a mirroring from before, where we have the putamen body, or putamen tail with the anterior cingulate. But what you're seeing is that as we see greater impairment in social motivation, there is a reduction in functional connectivity. So that's functional connectivity on the left and social motivation score. And remember that greater scores mean more impairment for both of these regions. The exception to that is with the inferior frontal and middle frontal gyrus, which has actually got an inverse relationship. And again, a mirroring that you would see before from the somatomotor results, suggesting that the group is actually giving us something of that similar information. You see that as stereotype behavior scores increase, so the more intense and pervasive the behaviors are, the greater functional connectivity there is between the putamen and the inferior frontal gyrus and orbital frontal cortex demarcated in this image. Frontal parietal is a little different from what we saw before. There's not quite as many, but we do see that as an individual shows more severe compulsive behaviors, there is less connectivity between the putamen tail and the inferior parietal lobule, with, again, that inverse relationship in frontal parietal network 13, where you're seeing a reduction, that as the compulsive behaviors become more persistent, you see greater functional connectivity from the caudate body to the IPL and the inferior frontal gyrus from the caudate. Now, the structural correlations or the diffusion results aren't anywhere near as interesting as we thought they would be. So for generalized fractional anisotropy, primarily what we saw was limited to default mode, and it was primarily associated with increases in generalized fractional anisotropy being correlated with increases in functional connectivity. Quantitative, on the other hand, had some slightly more interesting results. So there was actually primarily negative correlations between our quantitative anisotropy values and functional connectivity. This one is in the ventral attention network, so if you'll remember, that's that bottom up where you're aiming to certain things. And you see that a similar relationship in frontal parietal 8, where you see decreased anisotropy associated with greater functional connectivity between these regions. But something that, and again here, these are almost entirely subcortical frontal as far as our structural results go. and same for the other default region between the IPL and the putamen. What's interesting about these correlations is that absolutely none of them overlapped with any of our group or behavioral analyses, so these are unique unto themselves and could not be related back to any of the behaviors or contrasts that we saw previously. So as far as what this all means, we didn't see any alterations within these limbic reward regions. It was almost entirely limited to default mode or cognitive control or attention regions, suggesting that as far as the literature goes, maybe we're not seeing this general dysfunction in reward circuitry that social motivation hypothesis of autism has put forward. And we do see some trends of striatal cortical increases in somatomotor and default mode regions, but we also see these decreases in functional connectivity between putamen and precentral and default mode with anterior cingulate in dorsal attention networks, which is relatively novel. As far as the implications of that, when we looked at the behavioral correlations, the vast majority of them could be brought back to our original group contrast, suggesting that it's giving us similar information. But we also identified some caudal frontal and caudal parietal functional connectivity that could be related back to compulsive behaviors or social motivation that were not themselves identified in the group contrast. But as far as our anisotropy correlations go, we found very few, and most of them were limited to the caudate head and default mode and frontal parietal regions. With normalized quantitative anisotropy, we did find a few more results, primarily in cutaneal frontal and caudal frontal, and frontal parietal and default regions. But these were almost exclusively negative correlations, and since this is a density function, we think that it might relate to less dense fibers being more functionally connected, which goes with some of that apoptosis theory that's been out there, but we're going to need a lot more experiments to verify before I can make that claim. But to sort of summarize everything that I'm talking about, we didn't see a lot of our initial hypotheses that we were expecting to see, especially when it comes to reward and social motivation. So at least according to the social motivation hypothesis, it doesn't seem to be a general dysfunction in reward within this framework. But the weaker connections that we saw in a lot of the dorsal attention networks, which again is that focused attention, may suggest that it's playing a much greater role in this hypothesis than, or even in the pathology, than we're giving it credit. And we see those increases in subcortical to cortical functional connectivity with increasing impairment, especially in frontal parietal regions, may reflect this difficulty with inhibitory control in individuals on the spectrum, and we'd like to explore that more. So there are some definite conceptual limitations to this experiment that I did. We're going to need a lot more CAS data to assess the validity of a lot of these behaviors. Parcellation that I use doesn't include the hippocampus and amygdala, which is actually really, really important in that circuitry, and I'm looking forward to seeing where the work goes linking them all together. We would have liked to have had a lot more behavioral data, especially comorbidity, within the ASD sample, but unfortunately SDSU didn't collect comorbid conditions. We have a relatively low sample for the analysis, and just to put forward, a lot of the results that I showed you don't survive that FPR correction, so this is all at an uncorrected level. But I think if we explored it more with a little more power and some more nuanced analyses, we would have a better idea of how this works. The data that we used was only single-shell diffusion-weighted imaging, and if you're going to do subcortical analyses, multi-shell and diffusion spectrum is a lot better for getting that resolution back. There is a potential effect of distance dependence, especially within the sample, because I have a metric for that. And currently, we don't have any atlases or templates for developmental populations, but I think that's a great avenue of research. But the big one is that conceptually, as far as the structure-function relationship that I analyzed, that's a very binary level. So between two ROIs, what is the tractography result? That's not exactly how the brain works. We have multiple regions connected and moderated and mediated by the connections in between. And that's going to require a more nuanced analysis. something like a data fusion blind source separation or even persistent homology, both of which I'm very interested in pursuing here. But I want to leave you with the following hypothetical, because this is what's been bothering me for probably 8 years now. And I would like to share it with other people in the hopes that it will bother them as well. So let's say that we have all these different scales here: social motivation, communication, and the different repetitive behaviors I'm talking about. And we have a typically developing individual who displays none of these things, has a composite score across all of them of, say, 0. But then let's take patient number 1, who is a patient on the spectrum. That's sort of that classic patient on the spectrum. They have a mild to moderate presentation of all these different symptoms, and they have a composite score on their total behavioral ASD measure of 8. But then we have patient number 2, who's got comorbid generalized anxiety disorder, and as a result of that is not highly socially motivated, and may not have developed great communication skills because of it, but their incidence of repetitive or compulsive behaviors is actually really, really low. But this person will display the exact same global score on a diagnostic measure. And then we have, say, patient number 3, who is also on the spectrum, but has a comorbid diagnosis of attention deficit hyperactive disorder and obsessive compulsive disorder. Now, this person has a lot of repetitive behavior and compulsive behaviors, and maybe as a result of the ADHD, hasn't developed great communication skills due to the orienting problems, but is highly socially motivated, wants to meet people, wants to go out, wants to do things, wants to talk, just has this small deficit here. This person will also have a total composite score of 8. And the question that I want to present to you is, do we think that all 3 of those people on the spectrum are going to share the same functional connectivity profile? So just as a brief aside, I want to thank the Abide database and INDI for hosting the data, all the different developers for allowing me to use the packages and the analysis, a bunch of open source learning platforms such as DataCamp, Coursera, and even one I took remotely at University of Eindhoven Technology to improve my skills. Lyndon Parks' GitHub page, who provided the base code that I used for the distance dependence artifact. And you can look at any of my scripts or results as I'm posting them on the Center for Open Science just by scanning this QR code here. And at that point, I'll take questions.